many of the disorders, we don't have good medicine at all.
Mm-hmm.
So that really inspired me to say, I told myself, I cannot do this job. I have to go to research to find a treatment for pediatric disorders, basically. Because you're basically fight against time.
Yeah.
I told people, kids are growing up every day. They cannot stop growing development and wait for your development. You have to do as fast as possible, because the earlier you get into them, the better will be, the more effective will be. Use the SHANK3 study, and now it's into clinical trial, and it's very fast, because decided to license it about 3 years ago, right? And now it's in clinical trial. You don't see this kind of thing so happen quickly without a lot of support. But I think it's very promising in the future that gene therapy will be very simple. Go into clinic, have an IV injection, and go home.
Fix whatever you need to get fixed.
So that's our hope.
Welcome. I'm Ron Kleiman, and this is Generation Hope. Before we jump into today's guest, I want to share a quick snapshot of where genetic medicine stands right now in April 2026. Honestly, it's hard to keep up. Every week there's news. New trials launching, FDA designations being awarded, therapies moving from the lab into patients, and AI accelerating drug discovery faster than most of us imagined even a few years ago. Good example, Eli Lilly and NVIDIA have announced a major partnership to build an AI co-innovation lab aimed at speeding up how new medicines are discovered and developed. Gene therapies and RNA-based approaches, including ASOs, are no longer just someday science. They're in human trials right now, showing real promise for conditions that historically have had no treatment options, including Rett syndrome, Angelman syndrome, Phelan-McDermid syndrome, just to name a few. There are hundreds of trials either about to start recruiting, actively recruiting, or already underway. And the next 3 to 5 years could reshape medicine in a way we haven't seen in generations. Newborn genomic screening is gaining momentum, and the long-term vision is clear. Identify these conditions early and treat early. You've also got organizations like Enlorum, working to make personalized ASO medicines possible even when a disorder is extremely rare. And we're seeing more parent-led foundations forming all the time, turning community urgency into real research. And recently, CNBC Cures held its first summit, focused on accelerating investment and reducing barriers that slow new treatments down. The optimism I feel about our kids' future isn't about wishful thinking. It's based on what's happening right now. Kids are improving now. Some are being treated in ways that would have sounded impossible not long ago. And this is just the beginning. With that in mind, I'm honored to introduce Dr. Guoping Feng, one of the world's leading neuroscientists and a big part of the momentum that's driving this field forward. Dr. Feng is a professor at MIT, an associate director and investigator at the McGovern Institute for Brain Research, the director of the Hockey Tan and K. Lisa Yang Center for Autism Research, and a leader at the Broad Institute. His lab is working on some of the most important challenges in neurodevelopmental disease, including Phelan-McDermid syndrome, Rett syndrome, SYNGAP1 and Dravet syndrome. And his team also developed the mini SHANK3 gene construct, now being licensed by Jaguar Gene Therapy in their JAG201 program, which has now dosed 5 patients with no treatment-related serious adverse events reported to date. In this conversation, we talk about the science behind gene therapy, the race against time for kids with developmental disorders, and why this moment may finally bring real hope to families. If conversations like this help, please subscribe, like, and share. It's the best way to support Generation Hope. And now, Dr. Guoping Feng.
I'm Guoping Feng. I'm a professor in neuroscience at the MIT. I'm affiliated with the McGovern Institute and also the Young Tan Collective at MIT, which focuses on research to understand the brain and understand the brain disorders, hopefully translate our research into therapeutics for patients.
Let's start at the beginning. Do you remember what your inspiration was to go into science?
Yeah, so I actually graduated from medical school in China. in Hangzhou. I don't know whether you know, very beautiful city in Hangzhou, China. And in the final year of medical school, when you're intern in the hospital, you have basically internal medicine, surgery, OBGYN, and pediatrics. So, each are very interesting, but I love kids, so I decided I want to be a pediatrician. So I spent a lot of time in the hospital to intern. And at the end, I realized it's really difficult to watch kids suffer and sometimes watch them die. It really, and at that time, or even now, many of the disorders, we don't have good medicine at all. So that really inspired me to say, I told myself, I cannot do this job. I have to go to research to find a treatment for pediatric disorders, basically. I was only 23 graduate from medical school. What really, really difficult you realize is developing medicine is really, really hard. And without understanding the disease, there's no way, right? You just try luck is not the way. So I end up doing, more and more basic research, starting from molecular biology, molecular genetics, and working in Drosophila, which is a fruit fly, really tiny fly, to understand the technologies and the molecular biology power. Then moving on to mouse model, that my passion did not change.
Yeah.
I thought when I finished my PhD, I came to the United States, did my PhD in SUNY at Buffalo, and very cold place. Really cold. But it's a beautiful place to do my work. And I decided I really want to continue my research into disease. So I love the genetics that you can testing to do genetic manipulations to understand the mechanism. So I decided, but I want a model which is closer to humans, so and at least a mammal. So at that time, only mouse can be genetic engineered.
Yeah.
So I chose a lab in Josh Sain's lab at Washington University in St. Louis, a great medical school, doing my postdoc. And so you can see that I always focus on how can I eventually, it might take a long, long time, how might eventually my research can benefit the patient and develop some treatment for disease. So that it's always my passion, still my passion. So my lab mostly focuses on studying severe autism spectrum disorders. And
PMS is Phelan-McDermid syndrome is one of the areas that you're very passionate about. What are some of the other neurological and genetic disorders that you're working on?
Yeah, so Phelan-McDermid syndrome with SHANK3 mutation is our life basically, right? But we do work on many other disorders affect the kids. For example, Rett syndrome, which affect girls very severe, and also other neurodevelopmental disorders. We also work on like SYNGAP1, which is also a very severe neurodevelopmental disorder. Dravet syndrome, also very, very severe neurodevelopmental disorder. So the idea is for these monogenic disorders, maybe we can develop really good gene therapy approaches really to have effective treatment. Some of it can be cures. So depending on the age and the gene therapy medicine. So that's we also work on some other, because many other severe disorders like ADHD and other neurodevelopmental disorder or autism spectrum are not caused by monogenic, not caused by single gene mutations. We study their circuits to understand what are the defects in the brain cause that particular behavior, then use the cutting edge technology, the new technologies, try to identify a specific target that can be used to develop drugs to help these kids. And
the process for doing that, is it manual? Is it researchers, is like an individual researcher doing an individual task, trying to, tease out information? Is it something that, AI is going to help speed up or find things that, just open up the pipeline to be able to, find that information quicker? Or
yeah, this is a good question. So for research, if you overall, for example, use the SHANK3 study, and now it's into clinical trial, and it's very fast because they decided to license it about 3 years ago, right? And now it's in clinical trial. You don't see this kind of thing so happen quickly without a lot of support. So the number one thing, financial support. Someone has been really willing to donate the funds, right? By applying grants, all this is very slow and a very small amount. So we are very fortunate. We have two incredible donors, made it all this possible. Lisa Yan and Hawk Tan. They set up 10 Young Center for Autism Research at MIT, donated very significant amount of money. Then when we did a couple of years of research, they realized, oh, there is a potential. If we put a resource in, you could move this faster into clinic. So they, again, donated money, set up another center called Yang Tan Molecular Therapeutic Center at MIT. So that allows us to really focus and push. Don't have to worry, where do I get the next grant to push? Do the most impactful thing.
Focus on the science, not the administration.
Exactly. And the second thing is you need really talented young people, really, right? So I cannot know, I used to be, but now I want to work day and night because you're basically fight against time. I told people, kids are growing up every day. They cannot stop growing development and wait for your development. You have to do as fast as possible because the earlier you get into them, the better will be, the more effective will be. So, SHANK3, a former postdoc, she put a lot of effort, and people before her, there was a graduate student, MIT graduate student, Karen May did the initial work. Then we have a lot of other people, and that's you need young people. I think these are really tough problems. How to attract the young people into science is really important, and it's just because they don't get paid very high. They have a long training, because you said you actually have to learn so many things before you can really think independently. But I told them, nothing is easy. And my postdoc advisor told me, nothing is easy. If it's easy, it's already been solved. You are here because it's not easy. So it takes a long time to really, a lot of knowledge to do these things. I think one of our major job is encourage and attract the young people to the field to continue to push, right, for the better medicine, next generation medicine. I think that then MIT is MIT is a quite a unique university, really not only support basic research, but also really supporting translational things and entrepreneurial things, try to commercialize the discovery so they can benefit the society. So you need all this together, make things happen. Of course, you need a really, really good partner from industry like Jaguar Gene Therapy. They are a group of people really know how to do this thing because they developed the first CNS gene therapy, right? Got approved by FDA and now saved thousands of people's kids' life.
Zolgensma.
Yeah. So that's kind of, you need everything together to really make one. It's not one lab or one university. You need so many people working together. But I really have to give credit to the young people are willing to, blindly listen to me or listen to other people, because now you look at, it might work really in humans. But 5 or 6 years ago, when you just had an idea, who is going to risk their career to try this thing, right? So that's kind of, so young people are generally much more, they don't worry about it. I have kids, I want to do this. This is exciting to me. I may make an impact. So I think that's kind of, you need this kind of passion, right? So, because if you always, what if it doesn't work? Then I'm really in trouble because I couldn't find a job, all this. Then everything become very, just not exciting, and you're not going for the biggest impact, research.
It's hard when you have to worry about, family at home, kids.
Exactly. I think, it's really young people they have the passion, they have the smartness, they have the, physical capability to do all these things. I think it's really exciting to watch all these people. But on the other hand, if you make one discovery that it turned out to be impactful to patients, it also motivates them. Say, hey, my research is not only about publish a paper in a top journal. My research actually can help people. I think it's both mutually beneficial. I think in that way.
So is it also did Zolgensma also license the technology from? No, that was different.
It was an independent, different. But they gained experience by developing this, so make their next development much easier, faster.
Yeah.
Can avoid a lot of mistakes. Right?
I wasn't expecting a gene therapy for human trials for Phelan-McDermid syndrome gene therapy. I wasn't expecting it for like another 3 years, 4 years.
Yeah.
So when they announced that, kind of around now to 2025 that they're going to have their first person dosed, it was it's what spurred me to do this.
I think teamwork, right? So just the many different levels that have to work really well, and because it's you're not only concerned about efficacy, you also concerned about safety. Everything has to be tested. That's also tell you the importance for non-human primate research that give us the confidence that they are safe and they are effective, and they give you a much better chance to succeed in humans.
So in the marmosets, in terms of safety, was there with the procedures? It was also done through ICV.
No, we did the IV injection.
IV injection
in there, yeah.
And there's no blood-brain barrier is not an issue with marmosets?
We collaborated with Ben Deverman at the Broad Institute. So he developed a new vector which can pass blood-brain barrier in marmoset.
In marmosets?
Yeah.
But not in humans yet?
Not in humans. So apparently now we learned, many people have learned that blood-brain barrier are quite species-specific. And you can understand because the BBB is, protect us from the environment. So each species living in a different environment. So they have some specializations that a AAV vector works in marmoset may not work in humans or not even in mice.
Yeah.
It's really interesting to learn. So now people have started to developing AAV vectors that can pass in human blood-brain barrier. So then in the future, gene therapy, you just go to
IV
clinic, go IV.
So I know of a company that's working on a gene therapy for STX-BP1.
Mm-hmm.
I think it's called Capsida and something.
Yeah, Capsida is
So
one of the companies developing IV and AAV vectors. There are quite a few companies now developing them.
But so a company like Jaguar is going down the ICV route, injection into the brain, and they're starting their trial in January.
Right.
6 months down the road, things are going well, and Capsida is starting their trial, and things are going well there. So is that something that now they own that technology, and they may license that technology out to a different company, or to do SHANK3 through their technology, or would Jaguar now license their technology and kind of work with SHANK3? Like, is there any, what happens when there's a better technology out there?
Yeah, you can combine them because one is the gene therapy itself, one is the capsid, right? Okay. The vector.
Yeah.
So you can easily combine them with the next generation because the gene therapy itself works. Your capsid has, there is a better capsid. You only need to do IV, right?
So you put the vector in the capsid.
Yeah, just like a small molecule.
Yeah.
Early days, you have to do IV injection. Later on, people say, oh, this is too much trouble. Let's make an oral pill. So it's the same thing, right? But the medicine itself is the same.
Yeah.
It's just make it orally available, right? So I think that either way, there are multiple different ways, and depends on how companies work with each other. And most of the vector, AAV companies, they are willing to basically license out to, And Jaguar, they have their own partner also developed the IV AAV vector.
Oh, really?
Yeah, it's presented at the this year's Cell and Gene Therapy meeting, American Association of Cell and Gene Therapy meeting. They actually, they're quite good. So they will also move into clinic. And there are other companies doing that too. So there are many in the future. That is the future. Of course, injecting into the brain has one advantage, is that you don't get the AAV massively into other tissues and you don't want to treat it, right? So the AAV vector, you really have to test. If I inject into IV, into blood, how many get into the brain and the rest going to everywhere else. Is that toxic or not? Like all these things, it always have pros and cons. But I think it's very promising in the future that gene therapy will be very simple. Go into clinic, have an IV injection, and go home.
Fix whatever you need to get fixed.
So that's our hope.
I'm low on SHANK3.
So I think that's the hope that it will happen, actually. So it will happen. So many talented people working on this technology. Now that come back to your question, where AI and machine learning help. Yes. So these are a lot of the vectors developed by AI-based protein design.
Right.
You can design them. You can reduce the number of things you have to test. You dramatically increase the speed. Same thing for our animal testing, where you automated the machine learning-based behavior tracking. So it's much more objective. And you see, did this therapy work or not? You don't have to use humans to
sit and watch.
Sit down and watch and score, right?
Yeah.
You have computer tell you.
Yeah.
Computer is not biased if you design well, because it doesn't have a stake in it. So it just very precisely tell you. And now the new technology, it actually make it much more precise. What kind of thing it can detect? A lot of things previously we think, this test can be used in animal for a particular thing, but sometimes these assumptions are not correct because we don't know what mice or monkey are thinking, right? But with a computer, it's unbiased, just recording everything, and based on your daily activity, tells you how this animal is different from the normal animal, right? So and then it can help us actually design clinical trials. What should you test in humans? So I think a lot of new AI and the machine learning technology can really help us to speed up the design, everything.
And being at MIT, you're just surrounded by people who are in that field.
Yeah, to speed up that, actually Lisa Yang and Hawk started another center, computational modeling center, so that can work with the other group. So now there are a total of 6 centers at MIT to work together.
Amazing.
You should talk to her. Yeah.
I would love to talk to her. Does she have a family with special needs?
So they have 2 kids with autism. And when she set up a center for autism research, and they didn't know what are the mutations. So they just want to support autism research, find the treatment, understand better, and find the treatment. Later on, we suggest, maybe you should, test your kids, test your kids. What are the mutations? Maybe we can do something. Surprisingly, SHANK3.
No way.
Yeah. One day she showed me, I said, no way. This is SHANK3. Actually, they have a kid, SHANK3. It's 30. He is 38 also, 38 years old.
38.
Yeah. But it is a SHANK3 mutation. So they are just as severe as the many kids I saw here. Yeah.
That's what it always surprised me, like the variance in severity of what SHANK3 could do. Yeah. My daughter Evie, she likes single point mutation, but I mean she did have some language, and she was a lot more functional before she had a regression, and that's another thing, right? There's SHANK3, and what is SHANK3, and what is a result of whatever happened during the regressive period. Yeah. If there was any other damage that happened or why it happened, I think I still have not found anybody who said, regression happens because. Right.
No. Yeah. We don't understand yet. So, but from her point of view, she has kid with autism, especially with severe, and she knows, then she has the resource. She knows what needs to be solved. So she's very focused and made it possible for this, right? If you don't have that experience, or you don't, then you may, not sure what is the best thing to do. You can still donate, money, but you don't have insight to. In her case, not only she has the resource to really strongly support, but she also understands that we need to focus on, whenever she see a hope, like there's a hope that this might work, and she could put more resources on. This is just incredible for us and for the community. She's very low key, but I think this is the kind of thing we need more people really, because if you think deeply, to me, it's a little sad because many of the monogenic severe neurodevelopmental disorder, if with the current technology, if we put enough resources, we can solve many of them. However, from the commercial point of view, bigger pharma, they don't want...
Because there's small numbers for
yeah,
them,
and
right?
it's hard to... You cannot blame them because they have to survive first, right?
Yeah.
So they are going for the common disorders. But the common disorders, for these, you can make the biggest impact to the society and to the family and to the patient because these neurodevelopmental disorder, they need lifelong care. Many of them can never live independently.
Yeah.
But we have the means to solve that problem. It just needs a tremendous amount of resource and the people to work on it, right?
Different political systems all around the world. We've got capitalism out in the West, but what about someplace like China, where, I mean, is the research is all government funded, I presume? Is that, do you know what kind of resources they're putting into?
Yeah, so in the US, it's still, from a research point of view, it's still leading the world, right? Because of the technology and everything. In the care system, I've been here for over 30 years, so I still visit my parents and my families often. But I think the system is, they have the health insurance covered for the whole country, like Europe, the same thing, right? So it still depends on how much the insurance or they are government insurance, right? How much the government insurance program is willing to pay. So in China, my understanding, in the last 5-10 years, they put a lot more effort into these monogenic disorders, neurodevelopmental disorders, and because that's make a very big impact, and you see it can work. So they are also very keen on developing these things for supporting and for like if developing US, they would allow to go into China and the insurance will cover, right? Like here, right? So that's kind of, I think the main question is not only research but the commercial development. Who is willing to pay for the commercial development, right? Because if bigger pharma is not willing to pay to put into money, and so should the government, NIH, have a consortium, and they have tried that, right, to put really effort into. In the last few years, we see this kind of from NIH and the government organizations try to encourage the preclinical development into the clinical trial, right? So that will de-risk a lot of things. Once into clinical stage, have some indication will work, then biotech and pharma are willing to do that. So I think there are hopes, and because it will be really sad because it's a technical problem. It's not a conceptual problem. Conceptual problems are much harder to solve. Technical problem, you put enough people on it, we will solve it, right? So you can see that if we do it right, there will be a lot of advancement in this field for the next 10 years.
So again, it's not a question of if, it's a question of when.
Yeah.
Whether it's 5 years or 15 years from now, where a child is born with Phelan-McDermid syndrome or Rett syndrome or STX-BP1, and they will get an injection or a pill, whatever it is, and that will resolve that issue.
Yeah.
Have you thought about what that will look like in terms of living in a world like that?
Yeah, I think, so basically, these are very, very severe, neurodevelopmental disorder, right? So if we can solve that, and for the majority of the patients, it will be incredible, right? So because, I love kids, so I have 2 kids. They are grown up now, but the joy of having kids, is unbelievable when you have your child. Then 2 or 3 years later, you realize they cannot go to school like other kids. It's unbelievably, I just cannot imagine the pain the parents will have, right? And then you have to worry about their long-term future, and not only the daily thing can really, really drain you, right? So I think if we can solve, I will be the happiest person. I just feel like, this is something that we can do, we should do. Yeah.
You're going to get a very good indicator in 6 months to a year. You're going to have a very good, feedback on that.
We'll continue to work on it. So I think this things, it's also really motivational to inspiring to the people, young people in the lab. We train graduate students, we train postdocs, we train all kind of people, right? They're mostly going to graduate school after the lab. And they change their mind and say, hey, I also want to do this kind of research. This can impact a lot of people. I think it's really fun. For us, it's not what I gain, what a paper I need to publish. It's just how we can make system work for the patient. I think medical training is as an important of my background. Although I never imagined I would be in medical school, but this is something I think really, follow you for the life, that your goal, your job is to help patients, right? To make them in whatever way you can to. I think it's a really exciting time, and you need if I started 30 years ago, it wouldn't work. The technology is not there, knowledge is not there, but now it's really good time. So for really solve some of the toughest problems in the world and the brain disorders. Yeah.
Part of this project is to really try to figure out what that's going to look like for the patients at the age of 18. What does that look like? What does that mean? It's never happened before in history. Yeah. You're in an 18-year-old body, and suddenly, you see the world around you without being overstimulated and, all the other things. Yeah. And what that looks like.
Yeah, I got a lot of questions like that from parents. It's actually a really good question. It's really tough to answer. I think
we won't know until it happens.
Yeah, but there are a lot of ways to train the brain, right? So you basically, you ease them in, right? Yeah. So it's not going to happen overnight. You really provide the training. And our brain is so plastic, unbelievable, right? So it's so plastic, you can learn things. I learned actually skiing when I was 50, moved to Boston. And
great.
Then you form the motor circle, right? Yeah. So you circuits, that our brains, even at old age, is very plastic, so they can be trained.
That's exactly it. They're starting kind of like a day one, but they're 18 or 24 or 52. We don't know yet exactly how how well this is going to work in an older population.
The new supporting system for these patients. Yeah. But you watch them, there's nothing happier than watch your kids now suddenly can learn this, can do this. Like, I'm going to spend all my time teaching this, my son or my daughter, right? So I think that would be an incredible feeling. Everybody would be like really, really excited.
Absolutely.
Yeah. But the foundation is really important, right? And there's, organization, because a lot of the families don't have the medical knowledge. So, and through this kind of meetings, the conference is not only educational, but their participation is critical. Mm-hmm. Because of this organization, Jaguar can do gene therapy much faster, initiate much earlier, because all the data is already there, right? If they have to start organize, try to recruiting, to find where are these patients, it would take them years. So this is incredible, not only good for the family, but also for the development of drug development in the future. The Parents Foundation, Orange Foundation and Initiative Foundation are incredibly helpful, because parents are very passionate. They want to find therapeutics and treatment for their kids. So it's really, I think, also lobbying Congress to make sure continue to support. Mm-hmm. Congress has a lot of priorities, but this is one of the things we can do so well, help so many people, right? So I think that's also Parents Foundation, really, really helpful. Yeah.
Do you know of any people in Congress that have, kids that are, SHANK3 or Rett?
I don't know personally, but I'm pretty sure there are.
There must be some.
There must be some, right? So either former or current, or I think, because it's one of the most common monogenic causes of severe ASD.
Yeah.
And but we did met a few congressmen this year to try to, convince them supporting neuroscience research, brain research. So there is a neuroscience caucus, Congressional Neuroscience Caucus. There are a few of the congressmen are either neurosurgeons or, or veterans with brain injuries before, so they are really supportive of brain research. So I think getting the message over there. There are many other things to consider, but this is probably one of the important things that the country can do for a lot of patients, a lot of families. Yeah. There are a lot of organizations like Foundation, but also Society for Neuroscience, doing all this, educational work and to try to help to support the research. It takes a long time for any research, right? So to make it to the clinical stage, long time. Yeah.
Right now, like AAV9, that, like Jaguar is licensing that from MIT.
Right. Broad and MIT.
Is Broad a separite? Is it part of MIT?
Broad is a separate. So it's called the Broad Institute of MIT and Harvard. So it's jointly established by MIT and Harvard, but they are independent, except financially independent. But they are professors has to be either MIT professors or Harvard professors. So I'm part of the Broad also.
Is the work on the AAV9 kind of completed, and you're working on new vectors that are, like, for example, are you working on like a new vector that can hold a larger size gene, for example?
So we mostly focusing on technologies or approach that to treat the disease, because there are other labs that are specialized in develop vectors. So
I see.
Yeah, we are using their vectors to deliver our system into the body to treat the disorder.
So your system is what? Is the mini gene?
The mini gene or genome editing, new technologies. How do you correct the mutation?
Gotcha.
Or what's the best way to correct, or make it safer? So that's kind of thing we're working on. So there are technologies there, but it cannot be used in human because it's not safe.
Right.
Can we make it safer so that we can use it in the human? For Rett syndrome, that's what we are doing. Can we make a genome editing method much safer so we can use, in animal models, we already know we can correct the mutation perfectly.
Like base editing?
Yeah, base editing, for example. But if it's there for all the time, it may cause off-target effect that cause a lethal problem, right? You don't want them added randomly. So we are trying to develop ways that do your job correctly, then leave, get out, right? So that's the technology is still missing, so we are developing them. And so that's an example what kind of thing we try to do to really develop what is the best way, and because for human use you want safe, simple, right, effective. All too complicated usually doesn't work too well.
Pick two of the three.
Yeah. So that's kind of thing we are trying to develop for individual disorders. That's the gene therapy aspect. But in my lab, we have another part is focusing on psychiatric problems. A lot of kids or young people have depression, schizophrenia, OCD. All these are in young people, right? So especially depression, very common. So with the new technology, we can really map what circuits is disrupted in this patient. And we used animal models to test, are there specific target that you can modulate that can correct the problem? If you can, then you can try to develop drugs for that target. And in the future, you can just take orally or other ways to modulate that circuit. So that's kind of thing we're doing a lot in the lab. Currently, we focus on autism, the polygenic autism. Yeah. If we can really solve this, polygenic, some of the key symptoms, it will make their life a lot better, right? Yeah. Because they are still high functioning. Yeah. They don't have intellectual disability, but they still have a lot of problems in their life. How to keep a job, anxiety, inflexibility, all this to cause their life really difficult. Yeah. If we can solve these problems, you don't need to do gene therapy for this kind of people, right? So that's kind of where we have some really good, candidate in that area. For depression, most of the time they are normal, but if you can get them out of the depressive mode, they are totally fine. Yeah. Right. So, but if deeply depressed, people commit suicide, all these things, and every year, almost every university, their young kids commit suicide because of depression, right? Including MIT and Harvard. So if we can solve that problem, you can say, there are over 1 million people in the world commit suicide every year, right? Yeah. So that's kind of dying of suicide.
In looking at that, have you run into anything about psilocybin depression?
For all these psychedelics, right?
Yeah.
The mechanism is not clear. So a lot of people try to figure out what is the target, what is the mechanism.
Yeah.
So that's how this new technology can use, right? And then you can test in animal models. So we do similar things, but we don't directly work on psychedelics. We have our more neuroscience-based approach to try to figure out what is the circuits involving depression, for example. And once you figure that out, then as a neuroscientist, we know how to modulate it. So we are in the process of doing that. Yeah.
Do you have a timeline horizon goal for it, or is just you guys are kind of just plowing ahead, just trying to get as much done as you can in as quickly a time as you can?
For the autism, right? So that's my focus. Others, it's hard to say whether it will be 5 years or 10 years. But for autism, since we have been working on so long, I really want to push them into clinic. So we already see some data from mouse models that works really well, right? So now we are going to move into monkey models. If it works, we have an advantage. We are one of the very, very few places that have monkey models for SHANK3, for example. If it works on SHANK3, at least we can start testing, develop, drugs that are for SHANK3 patients. If it works, then you expand into all other, populations of autism. So that's kind of a goal. I think in less than 5 years, we should have something into clinic. Yeah, I'm very optimistic because some of the things works so well in animal models and not only on the behavior, but on the circuits, imaging circuits, right? Brain circuits can be corrected. If I see that, and we already saw it, then I know there's a much better chance to be translated into humans at a circuit level.
Let's go back to Jaguar. So Jaguar is they're licensing so what are they licensing from Broad? Is it Broad or MIT?
Yeah, Broad and MIT together. Right. So they license this mini SHANK3.
Right? The mini gene. Okay, so how did you decide? So you decided the SHANK3, because the gene itself is too big, is it like 10 times the size that can fit inside of?
No, it's like 25% extra, bigger than.
So you have to remove 25%.
Yeah.
Okay.
We actually end up remove more than that. Yeah.
Okay.
So because you have to put other element in.
Right.
Right. So yeah.
So how do you figure out what's important and what's unimportant, or what just has unknown significance?
Yeah. So basically, when I was thinking that, I went to one of the neurodevelopment disorder meetings, and they brought a lot of kids. And when you see them so severe, you want to do something. So that time, I was thinking, if we can put a part of the SHANK3 back, it's better than nothing, because it's just, you might be helping them to a certain degree. Then you can figure out the next generation better. And of course, we always want to do as good as we can. So there are a lot of study by many other labs, including my lab, many other labs, pioneered the SHANK3 study. So we know which domains are important, because you can use bioinformatics to look at which domain, which part of the protein is conserved through the species. If it is conserved, it's important, because evolutionarily they don't change. That means I cannot change. If I can change, I'm in trouble. So that's kind of information. Then people have done a lot of deletion studies. Deleted this part, say, okay, what's missing after I deleted this part? So with the current information, we know which part is important. Then there are a few parts that are not conserved. That means the particular sequence is not important. And they maybe play other roles, maybe because you need to be flexible, and then you can change, right? So that's kind of, it's not like each point probably has some important use, but they are just not critical.
Right.
Then if you put the critical part together, at least you may not be completely cured, but you may dramatically improve patient life. Right. So that's the first generation we want to do, right? And the reason we choose that is because many of them are deletions. Larger deletions, there's no way to genome edit it, change that one nucleotide. It doesn't really help because there are so many patients that are deletion, SHANK3 deletions. So gene replacement seems like a way that you can cover a lot of patients. Yeah.
Instead of delivering a gene through one vector, splitting that gene in half and delivering it and then reassembling it with 2 vectors.
Yeah. The technology is already there.
You can do that?
Yeah, you can do that. Yeah.
Okay.
So we already tested in some of the other genes. The problem is you need both of them getting into the same cell.
Yes.
So if you don't, then you have a half the gene.
Yeah.
The half gene could be not only not functional, but it make it worse.
Right.
Because we call it dominant negative form. Because now you occupy a position, then the normal copy cannot go that place.
Right.
Then you actually make it worse than before.
Right.
So the question is, how do you get both of them into the same cell?
Yeah.
And currently, just by random chance, right? You make a AAV-A, AAV-B, you mix them, you inject them.
Yeah.
And then you're hoping each cell have 2. Yeah. But the chance depends on how frequently you get. Yeah. So right now, the amount of AAV can get into the cell is very small, right? If it's, let's say, 100%, then it's always 100%. Yeah. 90%, it's 81%. 50%, then if your efficiency for each is 50%, then you only get 25% cell get it right, the others are wrong. Yeah. So that's the problem. It's still the delivery problem. The technology with 2 vectors working together once you get into the cell is already there.
Okay.
It works in animals. It's just hard to get into human yet.
Yeah. Okay.
Yeah, people already work on it. Yeah.
Oh, that's fun.
That's already developed many, many years ago. Yeah.
In terms of base editing right now is dangerous because of off target and things like that. But can you do base editing? Can you take kind of outside the body, do base editing, and then re-inject kind of the cells back in? So
that's what the people have been using for blood cell related disease. Right? So you can do that. For brain, that's really difficult, right? You cannot get the brain cell out of the back. But base editing is getting safer. So people have developed the next generation or third generation, right? So it's getting safer. I think eventually it will be useful. One of the major issues is actually you can only edit one specific mutation, right? So, and for patients, there are mutations all over the place.
Right.
So you actually, you have to develop for each of these point mutations. So the question in the future, how do you combine them together to develop, can treat many, many different things in one cell. You develop one thing, can treat many, many patients with different mutations. Right now, you develop one, can only treat maybe 1 or 2 or 3 patients because, what is only few patients have the exact same mutation.
Right.
So that is the problem. So now they have other called primate editing, can treat a stretch, small stretch. It's still, increase more, but it's still not the whole gene. So
it would go in this small stretch, it would fix whatever needs to be.
Yeah. That's in the development, by Prime Medicine called, David Liu, he developed this technology, developed a company called the Prime Medicine from Broad.
It's a private company? It's a private. It's not it's not
initially it's a research from Broad Institute and Harvard. Now it's in a private company to develop a medicine. Yeah.
So do Broad and Harvard own part of that company, or does it licensing?
It's usually licensed. Yeah. Usually universities don't own part of the company. They may have some shares because as a license agreement.
Yeah.
That's all. Yeah. They don't get directly involved in the company.
So all the work that you've done at MIT over the years
belongs to MIT.
Belongs to MIT.
Or Broad, depending where I did my work. Then MIT and Broad, any company can negotiate license from Broad or MIT, and MIT will get a royalty or license fee.
Is there ever anything where the university shares some kind of agreement, or the researcher themselves own any kind of part of?
Yeah, so the researchers themselves can also say, I want to license this, just like as a company. I want to spin out a company. That's also very often.
the researcher will license it from the university, back from university, and continue working on it.
Even I develop it, it's not belong to me. It's
Right.
always belong to the university.
Gotcha.
Then I myself, if I want to start a company, I still need to license from to the company. And so the company can commercialize, develop them. Yeah. Yeah, so everything you do in the university, your work, belongs to the university, the intellectual property.
Gotcha. Jaguar noted they're the exclusive licensor.
Right.
So the licensing agreement that they worked out with MIT and Broad means that they're the only ones who can license this technology.
Yeah.
Is it that they're the only ones who can license this technology for SHANK3 or for just like for SHANK3?
For
mini
SHANK3. Yeah.
Would you want to take a minute to explain kind of what a vector is? Kind of what you just did down there, but yeah.
So for the SHANK3 gene therapy, basically we make a smaller version of the SHANK3 gene, so you can get into the brain. To get into the brain, you need to put it into a vector. We call it a vector. This vector is a harmless virus, and specifically engineered that can get into cells. Right. So these viruses exist in the environment. There are specific types that we found it's harmless and can get into cells and can be used for medically. So that's it has been Jaguar used AAV9. There are AAV 1, 2, 3, 4, 9, and many others variations, right? So they use 9 because 9 has been proven very safe and in hundreds of clinical trials for many different purposes. But AAV9 in humans, there is one disadvantage. It cannot pass the blood-brain barrier. So you have to inject into the brain, into the ventricle, which is has a liquid in your brain, and it's circulating. So once you inject it in the ventricle, because it's circulating, it can get into all over the brain. So that's this gene therapy. So you basically, to packaging the mini SHANK3, we develop it into AAV9 vector. Then you prepare very pure, high purity AAV vector, then inject it into the brain. You only do it once because AAV9 or AAV vectors have been shown can live in your cells harmlessly for a long, long time, like decade. People have shown, right? So it will be one-time shot, and then when the AAV get into the cell, it basically uses our cell's system start to express the gene. In this case is mini SHANK3. Once it expressed, the cell knows. Cell, when you have a gene expression, it will translate into a protein. That protein is functional and getting into synapse, make your neuron communicate normally, then you should be fixed. So that's kind of, cartoon. It's actually, it works really well in theory in animal models.
Models too, yeah, right. Yeah.
So not only in theory, in practice, but the only thing we're still lacking is testing humans. So I think the key problem is not whether this thing will work or not, because we already know the cell it will work. It's just how many cells you can get into human brain, and how many cells, correct the cell, do you need in the human brain to make a big impact.
When they're going in, they're going to remove a bit of the liquid that's there, because I'm guessing they can't add it because of pressure. So what is that liquid?
It's called it's a
brain fluid?
Yeah, brain fluid is a CSF.
Okay, and that's the same that goes down the
spinal fluid.
Okay.
Spinal tab is the same thing. You get it at
so if that liquid is removed, does the body regenerate it over time?
Yeah, it will regenerate. You basically remove the amount that you're going to inject back.
Right.
So you basically make an artificial CSF, similar
not just saline or anything like that.
A similar component, and then you can mix and inject it into.
And how many neuron cells does a brain have? Billion?
Human brain is about 80 billion to 100 billion cells.
80 to 100 billion cells.
Neurons.
There's
another about 100 billion of glial cells.
Right. And we're just, and this therapy is just going after the neurons.
Right. So SHANK3 mostly function in neurons. Yeah.
So in a perfect world, they would deliver 80 to 100 billion AAV9 vectors that go in and attach to each of those for somebody with an SMP mutation, for example, with one working. So it would attach to all those 100 billion neuron cells, and now those 100 billion neuron cells would now function normally.
Yeah.
Is that the ideal kind of goal? Or?
But right now we cannot do that. So in the monkey study, we only have about 15 to 20% neurons, and they already behave normal. The reason is neurons don't function independently.
Right.
Like they make connections, and they're very plastic. If they are, with certain neurons normal, they can make the circuits function almost normal, right? So, if you can get into 5 to 10% neuron in the brain, it will make an impact, right? Of course, more and better, but right now it's difficult to get into all neurons. We don't have a way to get into every neuron.
What's the challenge there in terms of why does it get into some neurons and not? Is it that there aren't enough vectors in the liquid, or
no?
Like how many vectors, for example, would be?
Right. So it's basically a balance between AAV will attach to cells immediately, then getting into. So if you put too much, you have a local very high, but does not distribute everywhere very evenly, right? So it's a diffusion into the different part of the brain, and also each AAV vector type, like AAV9, they have preference for some regions and other, more in other regions. So it's not uniform.
Gotcha.
So in the future, with the vector engineering, with IV, it will spread better because the small blood vessels goes everywhere. Even that is not blood vessel distributing in the brain in different regions is not uniform.
Right.
So you will also get some region more or some region less. So ideally, in the future, you just want to correct the mutation. Then it doesn't matter because there's no overexpression. You just correct the mutation. The expression is controlled by your endogenous, your internal cell system. So that's the future ideal situation. So right now, most of the vector can reach about 30 to 50% maybe, the new ones, but they have not been clinically tested.
In humans?
In humans, yeah.
Once a vector enters a cell, is it possible for another vector to enter the cell?
It's possible, as long as they don't have the antibodies. So it's a virus, so your body will generate antibody against it. Yeah. It's called a neutralizing antibody. As soon as see the same thing coming, it will just block it. Right, antibody will bind to it, and then it will destroy it. So now, but there are some vectors their antibodies are specific, so you don't interact. So you can use a new vector.
Right.
And then people are now developing also use vector engineering to try to reduce the immunogenicity, to reduce the
immune response.
Immune response, so you can do twice or 3 times. Yeah. So all these will be available in the near future. Yeah. People are already starting to develop them now.
This is, one of the concerns for parents who are, who
want to be the first one, right?
Who are thinking about, letting their children putting their children in trials early on.
Right.
If they get it now and they find out, a larger dose was needed, that they couldn't do it. But that, again, it's only a matter of time.
Yeah, it's only probably in the next few years we'll have this. They already published the data show that they can engineer that. There's all the AI protein design, all this helps actually.
It's like I won't be able to put this out fast enough. Well, I think, it's time for us to head out. But
okay, great.
Thank you so much. This was really appreciated.
I think the work you guys are doing is really important. Yeah.
Yeah, I hope so. I hope a lot of people, listen to this and watch this and just make them aware and kind of as optimistic and hopeful as I think we both are.
Yeah.
About the future.
Great.
Thank you so much.
Great.
Thank you.
All right. Great.
I'd like to thank Dr. Feng for speaking with me and his passion. Be sure to check out our other interviews with top researchers, doctors, and advocates working to bring therapies and ultimately cures to rare disorders. And if you found this interview helpful, please subscribe, like, and share. When you subscribe and turn on notifications, you'll get alerted each month when a new interview drops, along with updates from Generation Hope. Till next time, stay hopeful.